Okay, welcome. So in this lesson I want to talk about DNA replication. Okay, basically, so the word replication means to duplicate, to make into something else, right? So just to recap, remember that cells in our body, so cells in body that contain nuclei, okay, remember, the only exceptions are red blood cells, right, that contain nuclei. The cells in the body that contain nuclei have chromosomes, okay, made of DNA, right? So if I was to draw a cell here, cells have got a nucleus. In those in the nuclei, you've got X's, which are chromosomes. If you were to zoom in with a powerful microscope, you could see that chromosomes, right? So parts. If you were to zoom into a part of a chromosome, you'd see that it's made of DNA, right? And DNA, remember, has that double helix structure, as discovered by Watson and Crick. So basically, each cell contains exact copies of the chromosome that were in the original zygote. So I'm going to be talking about gametes now. Were in original zygote. Remember, zygote is that initial the starting point. So for when fertilization occurs with the sperm and the egg coming in, you get a zygote, right? So it's an egg basically. So each cell contains exact copies of the chromosome that were in the original zygote, right? So you get chromosomes from your mum and from your dad. Okay, that become you. So the zygote is you. So it's then possible to copy DNA molecules, right? Because chromosomes are made of DNA. So it's then possible to duplicate DNA molecules, and that's called replication, DNA replication, the copying of DNA molecules. Example is that of in the process of fertilization, right? Sperm and egg gives a zygote, and the zygote contains exact information that comes exactly from the two parents. Okay, what are the steps to replication? So here they are. Use a different color. So here are the steps for replication, DNA replication. Okay, let me start a new page actually. Okay, so here we go. Step number one. And you can think of this analogy as for when you're zipping and unzipping something, right? Like a bag or a pair of jeans, right? A zipper. So step one, the double helix strands separate. Okay, i.e. they zip open, if you want to think of that analogy. They zip open. So basically, in this case, we're talking about the parent DNA molecule, right? In the case of reproduction, so the parent DNA molecule starts to unzip. So if I was to draw this, here's what it would look like. Okay, so here's the double helix. Okay, the top part starts to unzip, so the base pairs start to come apart. So here's a G and here's a C. So they start to come apart, right? Let's say another C here, another G, and so on and so forth. These guys are still intact, these base pairs, but the uprights, the strands of the ladder, the DNA starts to unzip, right, like a zipper. Okay, that's step one. So it unzips. Step two. Okay, bases. Unpaired bases. Unpaired bases. So bases that are separated, right? Unpaired bases of each DNA strand. Each of these attached to bases floating around in the cell, floating in cell, right? So when the bases they're exposed, the unpaired bases, when they're exposed to the nucleus, right? There are individual nucleotides, right, that are not part of the DNA chain. Then they pair up with the exposed bases, and due to complementary base pairing, remember the rules, you eventually get pairing, right? So that's step number two. Now let me go on to step number three. Step number three, the final step. Okay, so bases pair up. Step number three, sugar and phosphate molecules bond with neighboring nucleotides. Okay, remember nucleotides are made of those chains, and new DNA strands form. Okay, and hence the replication gives two identical DNA strands. Okay, you got the base pairs, GCs, and whatnot. So you get two identical strands of DNA in replication. So that is DNA replication. Let me now talk about genes and how they differ in structure. Okay, let me use a different color. So genes and how they differ. Okay, remember, a gene is a segment of DNA, right? So if I was to draw a double helix, okay, a gene is basically just a section of it. That's a gene. It's basically it contains the genetic code, right? So genetic code contained in a gene, right? Obviously, because it's made of the base pairs contain information, right? And this genetic code specifies the chemical structure of a particular protein. Okay, remember I talked about in the previous lesson of things like valine, right? Valine is an amino acid. Valine is a building block. It's a building block of protein, right? So the protein shake that I take contains a mixture of proteins, or a mix of amino acids. Amino acids, when you clump them together, they make a protein, which is the protein powder that I take. So a valine, for example, is a building block of protein. Now, many of these, so many genes are duplicates, right? Because when you look at a DNA strand, a lot of them might have the same acid, the base pairs, right? So they might have GC, A, GC, and whatnot, and so you can get identical genes, right? So many of them are duplicates, and basically the DNA structure of a gene determines the arrangement of amino acids in a protein. That's all this is saying, right? So in terms of gene structure, the difference between genes is in the order of the bases and the length of the DNA strands, right? So that's how they differ, right? Difference between genes is in order of bases, right? So remember how I said that duplicates, a lot of them are duplicates, a lot of them are the same. Well, they're the same only if the base pairs are joining the same in the same manner, right? If they differ, then they're different. So the difference between genes is order of bases and also the length of the DNA strands, remember? So the length will be different, and so you get different genes. So therefore, each gene then has a specific genetic code for the instructions of the protein, okay, which has a specific characteristics or a function. For example, proteins such as collagen, right, which are found in ligaments and tendon, and keratin, right, the protein keratin is found in your skin, hair, and your nails. Okay, they're used for different things. So I'll repeat that again. So each gene has a specific genetic code for instructions of a protein, and that protein can have a different function that can be used for different things. Okay, so that's genes and how they differ. Now talk about proteins. Remember, proteins are made of amino acids. They're very important for muscles and tissues, right? Proteins, they perform two functions. Here are the functions of proteins. These are two very important functions. Number one, they make up a large part of cells, remember? Remember, cells are made of chromosomes. Chromosomes are made of DNA. DNA genes, they're made of amino acids, right? And amino acids are the building blocks of protein. So a large part of cells. Number two, they act as enzymes. Okay, enzymes are chemicals that control all other reactions, right, in cells. Okay, enzymes. So they're chemicals that control reactions in cells. Okay, and basically, as I mentioned, genes instruct a cell to put together a particular protein, right? So genes instruct cell on how to construct a protein. Example, collagen. Example of a protein is collagen, also keratin. Okay. And that's all I wanted to talk about this lesson. So I talked about DNA replication. You can think of it as a zipper, unzipping and zipping something. So the strands break apart, then the bases, the unpaired bases of each DNA strand attached to bases floating around in the cell, right, in the nucleus. And then you get two new DNA strands that are formed. I talked about how genes differ in their structure. Remember and how they contain the genetic code for making a protein. They differ in, say, the order of the bases and the length of the DNA strand. And I talked about proteins being important in cells and acting as enzymes.